Hello, everybody. Welcome to today's lecture. So we're going to continue talking about the coronavirus. So I hope you've been finding this week interesting. Today we're going to talk about how it infects our cells, what are the symptoms, and a little bit as to how it's treated as we go through. Okay? All right, so just a little reminders. Remember, the virus is the SARS-CoV-2. And the disease it causes is COVID-19, and that stands for Coronavirus Disease 2019. It's believed to have originated through animal to human transmission, so it's not completely understood at this time, but we think it probably came from bats. But now it's spreading via human to human transmission. Okay? And coronaviruses are a family of viruses. And so COVID-19 is related to two other diseases, SARS and MERS, which were previous coronavirus outbreaks that you may have heard of. Okay, so what we're going to talk about first is how does SARS-CoV-2 infect cells? As we go through these slides, I'm going to stop and ask you some questions along the way. So you want to have a pencil and some paper handy. We're going to try to jog your memories about some previous units as well as some of the things we've been learning so far. So you might have to look up some of the information, hopefully remember some. So let's see how you do. All right, so these illustrations were actually taken from the New York Times. They have a really good section on coronaviruses, so you can check that out. So here's the first one. So the first thing the virus has to do is get into the cell. If it can't get into the cell, it's not going to cause an infection. So here's our coronavirus. So remember, it's enveloped, so it has a cell membrane. And then there are proteins that are embedded in this viral membrane. Okay, and then here's the genetic material inside. Here is the cell it's going to infect. And in the case of coronaviruses, and this one in particular, it attaches to cells in the airways. So this is our respiratory tract, our lungs, bronchioles, things like that. So here's the cell membrane. And remember, our cell membranes also have proteins embedded in them. And one of the proteins in our respiratory tract is called ACE2. And it just so happens that the proteins in the viral membrane happen to be able to bind with this protein in our respiratory tract. If this didn't happen, if these two proteins didn't match up, then this coronavirus could not infect our cells. But it just so happens that these proteins line up, and so it's able then to bring the coronavirus into the cells. And that process is receptor-mediated endocytosis. Okay, so you may remember we talked about this way back in our cells unit. So your first question of the day is, what is receptor-mediated endocytosis and what do we use it for? Okay, once the virus is into the cell, it releases its viral DNA. Okay, so remember, in coronaviruses, that means it's going to be releasing RNA because they're RNA viruses. Okay, so questions 2 and 3, what does RNA stand for? And in human cells, what does RNA do? All right, so in our cells, once the virus has infected them, basically, remember, it hijacks the cell. So it uses our machinery to make proteins and to make more RNA molecules so that it can make more viruses. So the virus's genome is less than 30,000 genetic letters long. Remember, ours is 3 billion. So it's pretty small, all right? And so the infected cell can then read the RNA, and it uses that RNA to then make the viral proteins, okay? Just a note here about different types of microbes. So we've been talking about viruses. So those are different than bacteria. Remember, bacteria are cells. They're prokaryotes, and there's lots of diseases that are caused by bacteria as well. So when you have a bacterial infection, you can take an antibiotic. So if you've had strep throat, you probably took an antibiotic. All right, but antibiotics don't work against viruses. Viruses, we generally use antivirals, or we try to prevent the viruses from infecting by making a vaccine against the virus. Okay? All right, so our next question. All right, these are RNA particles, and we said that it's 30,000 genetic letters long. So what are the genetic letters in RNA? All right, and then the next question is, notice this little green organelle, all right, and notice it's reading the viral RNA and making that protein. So what organelle is this? Okay. Once the proteins are started to be made, right, the machinery of the cell can then process it and form more copies of the coronavirus. So question number 6 is, what organelle is this? All right, and what does it do? Okay, and then new copies of the virus are assembled and carried to the outer edges of the cell. All right, oops, I should have made this a question. So if they came into the cell via endocytosis, how do they exit the cell? Remember exocytosis? Okay, so that's a freebie. All right, and then that's how the infection is spread. So each infected cell can release millions of copies of the virus before the cell finally breaks down and dies. These viruses may infect nearby cells or end up in droplets that escape the lungs. So remember, these infect the respiratory tract. So every time you cough, or even sometimes like during talking, little bits can be spread out to either other surfaces or other people. Okay. So once our cells are infected, that's when the immune response is going to kick into gear to try to get rid of basically those infected cells and get rid of the viruses. Okay, so in later lessons next week, we'll talk in more detail about this immune response. But basically, this is what causes the fever. It's actually your immune system fighting to get rid of the virus. Okay, and most of the time, this is pretty effective. But in severe cases, the immune system actually overreacts and starts attacking lung cells. And this causes the lungs to become filled with fluid. And that's how some of these patients are actually developing symptoms of pneumonia because their lungs are becoming impaired. All right, and then as we just said, coughing and sneezing can expel virus-laden droplets onto nearby people and surfaces. All right, and the virus can remain infectious for several hours to several days. It depends a little bit on the surfaces. The virus can live longer on certain surfaces than others. Infected people can avoid spreading the virus by wearing a mask, but healthy people do not need to wear a mask unless they are caring for a sick person. So as long as you're staying that social distance away, you should be good. And then washing your hands to make sure you're not picking it up from any infected surfaces. All right, so what are the symptoms for COVID-19? So there are actually a lot of symptoms. Go to the CDC website if you want more information. These are the 3 most common: fever, cough, shortness of breath. And notice they can occur 2 to 14 days after exposure. And this is one of the reasons why it's hard to prevent the spread, because sometimes people can spread the virus before they even know they have it. Okay? So as I said, fever, cough, shortness of breath are the most common symptoms. Okay. And in some people, so these are people that it becomes more serious, and these are some symptoms that you would want to get medical attention immediately, is if you have difficulty breathing, shortness of breath, pain or pressure in the chest, confusion or inability to arouse, so inability to kind of wake up and be alert, and bluish lips or face. So that would indicate you're not getting enough oxygen. All right, again, this is not an all-inclusive. I'm just sort of giving you a little bit of information. Check out the news or the CDC website if you really want to know what all the symptoms are. All right, and here's what's been happening in the U.S. So you can see California, Washington, and New York are the states hardest hit right now. And so these states, it kind of spread pretty quickly before they really took a lot of measures. But because it started here, it allowed a lot of these other states, including us, to try to take these really more severe measures because we want to prevent this spread that's being seen in other states. Okay? All right. And then just a short note, you've probably all heard about testing for the coronavirus. So the test that they're using is called polymerase chain reaction, or PCR. And basically what this does is they collect the sample, and then they take any RNA in that sample and they amplify it. So this is what this polymerase chain reaction does. And then once you have it amplified, you can actually study it and see if it matches the RNA in the coronavirus. Okay? So, question number 7. Do you remember, we just said this earlier, what is the genetic material in the coronavirus? So what are they amplifying and comparing? All right, you've all probably heard at this point about flattening the curve, okay? So what we're trying to do here is this is time since first case versus number of cases. What we want to try to do by all these precautions we're taking is spread out when the cases are arising so that they're all not happening at the same time. And this will ease how the healthcare system can deal with it so that we don't overburden the system. Okay? All right, science, math, real-life problem solving. Okay? So you've all heard a lot about social distancing. And so what social distancing is aiming to do is flattening the curve. Okay? So we're going to look at some graphs. So we're going to see people who are healthy, people who are sick, and people who are recovered. And they're going to be these color-coded. All right? And then we're going to look at the time lapse. And we're going to look at 3 different situations. Okay? So these are from the Washington Post. So the first one is if society kept its normal behavior. So if we didn't do all the things we're doing, what would happen? And you notice we get this huge curve, right, where we get a lot of sick people and in a short amount of time. And this really overwhelms the healthcare systems. Some other countries, so Italy, for instance, has been seeing this happen where they really overwhelm their healthcare system and were unable to deal effectively or as efficiently as possible with all the people that got sick, which means maybe some people died that could have otherwise been taken care of. All right. Situation number 2 is some people self-quarantine and there's some limit of social interactions. So now we can see that curve has been brought down and sort of broadened. So now we're not seeing all the cases all at once, but there's still a substantial amount of sick people. Okay? So this helps because, again, the hospitals don't have to deal with them all at the same time. The best case scenario is what we're aiming for now. So people self-quarantine and limit their social interactions. So now we really flatten that curve out so we can actually prevent some people from getting sick, all right? And then the people that do get sick can get the best quality of care. So that's what we're aiming for. All right, so here are your last 3 questions. What is social distancing? What is the reasoning behind social distancing? Okay, and then what I want you to do is just sort of reflect on this week's lesson. I know a lot's been going on. It's weird adjusting to this online method of learning. So what did you find most interesting or helpful? So that could be in my lessons, or maybe even tell me about some other things you've been learning in other classes. How are you coping with the online lessons? What's easy, what's hard? What would you like more of? So you've been experiencing a lot of different things in all your classes. What do you think's worked really well? And what do you think maybe hasn't worked as well online? Okay, so what should I plan for future lessons? Okay, next week we're going to finish up. We're going to talk a little bit about the immune system and the respiratory system, so you can learn a little bit about that. And then we'll talk about vaccines. Okay, do your questions and then have a great weekend.